Hi, thank you all for joining today. My name is Jakob Woodall. I am a master's student at San Francisco State University in the Amphibian Conservation Lab working under Dr. Vance Friedenberg, and I'm going to be presenting my research today for my master's project. So the disease we'll be discussing today is called chytrid or amphibian fungal disease, clinically known as chytridiomycosis. It is caused by the fungus Betrochytrium dendrobatitis, or fortunately also referred to as BD, which is much simpler to say. BD is an aquatic fungus, so the zoospores, which is this, oh, the zoospores, which is this stage, is the mobile stage and can be transported aquatically or through skin-to-skin contact. BD feeds on the skin of amphibians and ultimately can cause mortality due to preventing electrolyte transport across the skin. It has been linked to declines and extinctions globally in North America, Central America, South America, Europe, Australia, and in California, it's been linked to the local extirpations of several frog species in the Sierra Nevada region. The species I'll be discussing today is my personal favorite frog species in California. It is the western spadefoot. They are Spia himandai. It is a California endemic species being found in the Great Valley down the south central coast. Recent work has identified two distinct clades in the species. There's a northern clade and a southern clade that's divided by the transverse range. It has been recently proposed to be listed as threatened under the Endangered Species Act and has been petitioned by the Center for Biological Diversity to be listed as threatened under the California Endangered Species Act. So hopefully there'll be more protections for it soon. It also is a species of special concern in the state. It is understudied, particularly the adult life stage or post-metamorphic life stages. Much of the previous work in the species has focused on the larval life stage. This family Scapiopodidae is unique in that they have the shortest larval stage of any continental North American amphibian. The western spadefoot can metamorphose in 35 days from egg to froglet, which is extremely fast for a species in our area. And following in this understudied portion, much of the previous work for the species has also been concentrated in Southern California. Prior BD sampling in the species has been done by the USGS, again in the southern clade, and they have found infection prevalences of 0 to 31% at sites. At sites that are BD positive, they have found prevalences from 7 to 31%. And prevalence is just the percent of infected individuals in the population or in the sample. And here's a range map for the species to kind of orient you about where we're talking about. This red line is my approximation of the transverse range, and the gold star is my study site that, where I've conducted all my research that I'll be discussing more. And it is Carnegie State Vehicular Recreation Area, which many people are probably familiar with. It is a off-highway vehicle recreation area managed by the California State Parks. It has a very, very rich history, both in the park and in the Crow Hollow Valley, both naturally and anthropologically. It's the intersection between the Diablo Range and the San Joaquin Desert. And there's been many, many, many generations of herpers to cruise this road looking for all sorts of amphibians and snakes in the spring. Prior studies by the park scientists at the site have looked at BD and California red-legged frogs, the movement of western spadefoots, and at spadefoot larval morph plasticity. The BD study in the park was part of a larger genetic population study of California red-legged frogs in Central California. They tested 18 red-legged frogs in the park and found 12 of them positive for BD. Their infection intensity, we report that for BD as a zoospore equivalent, so the approximate amount of zoospores that are on the frog based on the DNA fragments that we're testing for. And they found ranges from 2 zoospores up to 4,200 zoospores. And generally, for other species that we've measured in, we start to see mortality in certain other species at about the 10,000 zoospore level. So for my project, I was interested in investigating the BD infection prevalence and intensity in the northern population of the western spadefoot. There's a historical component to my project in which I went and I swabbed all the available museum specimens, looking at the post-metamorphic life stages, so the froglets, juveniles, and adults, and then a contemporary component where I am looking at prevalence and infection intensity, looking for differences between life stages for metamorphs, juveniles, and adults. Previous work in our lab for other species has shown that metamorphs generally have the highest infection prevalence and infection intensities. And I was also interested in if there's any seasonal trends. Spadefoots are a very terrestrial activity limited species. So they have a very defined terrestrial activity peak in the spring, and then they'll head down into the burrows where they may or may not emerge depending on climatic conditions until the winter. However, as many master's students and PhD students will tell you, projects never go to plan. Unfortunately, last year, it was a very poor rain year at my site, and we had no successful metamorphosis. We did see successful reproduction, but the pools that the spadefoots reproduced in dried out before they could metamorphose out. So I already kind of lost some of the life stage data, and there are only juveniles and adults present, but we still had the seasonal trends to look at. And so here's a picture of the two of the western spadefoots from our site. On the left, we have a juvenile frog, and on the right, we have an adult frog. Previous work looking at a histological study that looked at the gonad development in the species had found that adults are generally 37 millimeters or greater in snout urocell length or snout vent length. For our sampling, the sampling for the museum specimens and the living frogs is the same. We swab them on their ventral side, doing 10 swipes on the stomach, 5 swipes on the inside of each thigh, and then 5 swipes on the back feet. We target these areas because these are the areas that are most likely to come in contact with the other frog species if they're amplexing each other, and they're also the areas that are most in contact with water when they're active in breeding. And it's where we typically are finding the infections. I am a bit of a busybody, so I also ran several side projects at our site because I couldn't help myself. To learn when we should be targeting these surveys, we installed some passive autonomous recording units to conduct passive acoustic monitoring to figure out when they were starting to breed. I also did a separate study where I conducted time series surveys to determine when we should be surveying in a given night for a frog. So these were overnight surveys where I went out once a month and surveyed 3 times spaced evenly across the night starting an hour after sunset, which were very exhausting but very illuminating. Because we were collecting morphometric data to help us identify the adults, and it's just standard data to collect as we're handling the individuals, I've also, using a paper, developed a scaled mass index for the species to look at body condition across time, like what trends we might be seeing. And then because we're doing these standardized surveys, I've also developed an environmental covariate of terrestrial activity model to see when we should be doing these surveys for the species, especially going forward, if they get listed, when people should be looking for adults. Most of the presence-absence surveys for the species focuses on the larval stage. Larvae are very contained, obviously, to water bodies. They're not going to be dispersed through the landscape. However, as we found out at our site and other sites, they don't breed every year. So if you are trying to do these targeted larval surveys and it's a non-breeding year, you might miss the presence of the species. And this is, sorry, I should have added. This is our audio moth recording unit, the autonomous recording unit that we were using for the acoustic studies, up close version, and then here's it deployed in the field. My coworker who does this a lot with birds advised on the weather protection for it, and we found that this double bagged method with desiccants is both cheap and works great. For my historic methods, I visited 2 local institutions, the Museum of Vertebrate Zoology at Berkeley and the California Academy of Sciences. Fortunately for me, the vast majority of the post-metamorphic northern clade western spadefoots in collections are at these institutions, which was a breeze then to sample. And we took the frogs out of jars and swabbed them as we would for the live frogs. And here is my team of undergraduate researchers diligently at work swabbing at the Museum of Herpetozoology. This project would not have been possible without their help. For my contemporary study, we conducted nighttime visual encounter surveys, which is a standard amphibian survey approach where you just go out with a flashlight held at eye level, and the frog's eyes will reflect back the light to you, so it makes it easy to find them. We recorded environmental conditions at the start of surveys, captured frogs in containers to avoid potential contamination, and then set up an in situ processing station where we swabbed them and recorded the morphometric data, the physical data. So the snout urostyle length, gape width, which is the widest part of the mouth, and their weight. And here's an example of, again, the sampling and those measurements. The gape width was a sort of QA/QC measurement. It's highly positively correlated with the snout urostyle length. So by taking both measurements, if something was recorded wrong with weight or with one of them, it was easy to identify in the data entry step. And here's our team. Our in situ processing station was the back of my car, which worked wonderfully. Back in the lab, we extracted the DNA from these swabs, and we ran a quantitative PCR to determine the presence of disease and to determine the amount of the zoospores present. And it's just a really fancy way of saying we are targeting a certain region on the chytrid fungus that is highly conserved and isn't found on other fungus species. And when we find that region, our samples, it attaches a dye to it. So the machine reads the amount of fluorescence from that dye to determine the how much of it is present. Unfortunately, the results were great for the frogs, but devastating for me personally. I've tested 77% of the historic swabs so far, and all of them have tested negative for the disease. Fantastic for them. There are two terrible type of results to get in the laboratory setting, all positives and all negatives. With both of them, you then have to do a lot more work to ensure that your results are accurate. And so for ours, we run positive and negative controls on all of our plates to help to let us know if we have contamination or if our reagents are acting appropriately. And then so this shows the number of frogs present in collections from each of the counties. There's a major skew in collections for the species, largely found in the San Benito, San Joaquin, and Monterey. Or Madera, sorry. And then this is the same data, but looking at the decades that the specimens were collected. Prior work in our lab has found that chytrid in this area generally starts to show up in the 1960s and will rise in infection prevalence through like the 70s and the 80s. Unfortunately for me, or unfortunately for frogs, they just had 0 infections. And here is the results from the contemporary swabbing. Again, all these frogs at our site tested negative for the disease, which is, again, wonderful for them. To check on these results, we also swabbed western toads, Syrian tree frogs, and California newts present at the site with the spadefoots, and they all also tested negative for the disease. The previous, yeah, there's something going on there. The previous work, again, with the red-legged frogs, was focused on sediment basins where the frogs are found. The water from those sediment basins flows into Crowell Hollow Creek, where we conducted our studies. So in theory, and the red-legged frogs are also found in the creek. We're not sure what's going on. And talking about the seasonality of the terrestrial activity, you can see here, most of our frogs are found in spring. We did survey through this time period. We just had no frogs. And again, here the results, 100% of the contemporary sampling has been run. over 266 swabs were taken from Spadefoots, all negative. 167 swabs were taken from the museum specimens, all negative so far, with 78% tested. Which, yeah, and this is me after doing plate after plate after plate. This is one of our favorite frogs from the collections. Now for the other studies. For the acoustics, we ran into an error with the recording schedule. We set the timer for the recorder on the wrong time zone. So they were recording earlier than we intended. And as such, we didn't really get great data from 3 of them. We tried to target our recording placement at areas where the frogs had been documented breeding the most in the park. Again, we did not see very much reproduction in the park, and the areas where we did see the reproduction was actually areas that they historically have had the lowest, the least amount of breeding activity. So we sort of missed all of our windows, and of the 2 remaining recorders that were in the proper area, they had chorus frog leks set up underneath them, which are very loud and work to drown out most of the other sounds. And then otherwise, Tesla Road, Corral Hollow Road is very, very active at all times of the night. And so the vehicle noise also made the data processing a little more difficult too. And here's one of our culprits. This is a Syrian chorus frog or Pacific chorus frog calling from the creek habitat. Now for the time series surveys. The first survey in January did not detect any frogs. It was too cold on that night. But the following surveys were successful, and I analyzed the results with just a simple ANOVA to look at if there's any differences in trials or month. Trials was not significant. So trials, again, are the 3 surveys spaced over the course of a night, starting an hour after sunset, and then, depending on how long the night was, every 4 to 3 hours, or 3 to 4 hours after. Month, however, was significant with the highest number of frogs, which matched our data observed in March. And so here's that data. It seems like it would be significant in these surveys, but it ended up not being so. And this was great because prior work with their currents has not looked at when they're active at night. And so there was kind of a lot of rumors present about when we should be doing the surveys and if frogs were most active like an hour after sunset, if you could go out 4 hours after sunset. And so the time series showed that if they're going to be terrestrially active that night, you can kind of go out whenever. I think my latest survey was at 3 or 4 a.m. and there's frogs present. For the scaled mass index, I followed the methods outlined in this wonderful paper by Pieg and Green in 2009. Previous body condition indexes just generally look at body mass divided by like length or snout urostyle length for our case. They had I'm not going to get super into the arithmetic behind it, but they had a different way to kind of create a scale for the different size classes that you might see. And it worked great with the data we were already collecting at the site. We found, which was super interesting, there's clear clustering between the life stages, with adult spadefoots again being greater than 37 millimeters snout urostyle length. Clear clustering in these scores between the juveniles and adults. There were seasonal trends present, and we had 3 frogs that scored greater than 20 on the index. All were females in late March and early April, when we would expect them to be potentially provisioning eggs, which is really interesting. And maybe this is something we're hoping to get more repetitions with and see if that's something we can use to identify females that might be doing that. And here is a graph of that result. So in the red are the adult frogs, and the green are the juvenile frogs. On the x-axis is the score on the index, running from 0 to 30, and on the y-axis is the frequency of that score for our data set. And so there's just very clear clustering of the data, and we're not really sure why we're not seeing any like in between. It might be due to when we're seeing the juveniles and adults, there's that period where we're not really finding them. Sorry, I think I'm running out of time. For the juveniles, we saw a clear increase in body condition over the course of the active period from February to May. For the adults, they were pretty similar in condition throughout the season. This is not using a linear model. This is using a low S model, which looks more at the trends present in the data and isn't really good for predicting out. There is this bump, though, in female condition present in that, again, that late March, early May period. For the environmental covariate study, it's a lot to go on here. I used a general additive model, which is better used for biological data. I had a lot of environmental predictors to start. Ultimately, starting air temperature was the only significant predictor with a nonlinear relationship. So there's kind of a peak of abundance with the temperature, and they prefer between 7 and 20 Celsius or 45 and 65 Fahrenheit. And days since last rain ended up being a non-significant, slightly negative association. And here is one of our emerging frogs. Again, thank you very much to Save Mount Diablo Foundation for the funding for this study, the ARC's Northern California chapter also for scholarship funding, and for Stillwater Sciences, a local environmental consulting firm for lending their autonomous recording units. Thank you to all these wonderful, wonderful individuals for their assistance with the study, including my committee members, our director for our genomics facility at State, my wonderful, wonderful undergraduate researchers. This study would not have been possible without their support and help in the field and in the laboratory setting. Elizabeth Getberlet and all the wonderful Carnegie Park staff and Diablo Range District staff who made this project possible, and the several other wonderful individuals who've offered assistance, a lot of assistance with the project. And for all my field volunteers, I've had a lot of field volunteers with this project. It's a lot of data to collect. I've had close to 100 individual volunteers come out across different nights as we were surveying weekly through that period, which I forgot to add. Thank you.
Mr. Time, if you have questions for Jacob, please leave them for lunchtime. Thank you, Jacob.